use a mic. Can everybody hear me okay? Yeah. Okay, great. If I need to hold it closer, just tell me. Okay, so everything that we've talked about in this sort of genomics session so far will hopefully click with what I'm going to show you. It's going to echo a lot of things that Moni talked about. It's going to echo a lot of things that the data management presentation we just had talked about. But I'm going to talk specifically about one genome browser. And they're actually, they are more than a browser. They actually annotate genomes. They provide gene models, called Ensembl. And this is something that you can start using right now because there are 2 Manakin genomes already in there. So I'm going to walk through some features that I think are very useful. I'm going to walk through some features that I think are going to help you get data faster. And things that I don't go over, I either don't know or I just didn't have time because this is a very big database. So, and I'm going to show you one way to get these to get to things. But much like a super interconnected website, there are 5 ways to get to any one page. So Ensembl is sort of circular in that sense. So just keep that in mind when you're navigating yourself. If you have internet, feel free to follow along. But anyway, Ensembl is a project between a European group that does a lot of bioinformatics, this huge conglomerate that hosts a lot of sites that I'm sure all of you have used at some point or another that deal with bioinformatics and the Sanger Institute. Let's skip over details. I'm gonna do a whirlwind tour, so please bear with me here. This is what the homepage looks like at ensembl.org. You have a sidebar over here. I just want to point out, it'll tell you what release the current release of this entire browser is. And this number is probably going to change if you're working with these genomes for more than a spell. And what you're going to interact with is are these search functions. And I'm going to show you how to get to the mannequins because that's what you all care about. There are 3 different ways you can get there. I'm going to show you how to get there using this link down here to just view all the species that Ensembl has. And after you type in filter, so I typed in Manakin, and now I can see that there are 2 Manakins in Ensembl's database. And that means that there is a genome for them, and that means that there is a gene build, or a set of gene models. And there are, in addition to gene models, there are transcript models, and there might even be protein models. So we're going to walk through what all these things look like. I'm going to try not to play favorites. I'm going to start out with the blue crown, and then I'll go to the golden collar because I don't want to offend anybody. So if we click on this blue collar mannequin, it takes us to the sort of landing page for this genome, and there's a lot of information here. But just to point out sort of the starting point, this more information will take you to something that looks very similar to an assembly page in NCBI, if you're familiar with that. And this tells you some background information about the genome itself. It gives you the accession number, so this is, you can type this in NCBI and find it there. And it tells you things about when this gene build or the gene model set was released, and the most recent one, and things like how many different kinds of genes are part of this set. So let's jump right into finding a gene of interest. I'm going to use the estrogen receptor 1 as my guinea pig today. And again, this is just back up the home page for this particular genome. Type it into the search bar and you go. You're going to get a search results page. It will have genes. You can see that here, the sort of last word here, gene, transcript, protein family. You can filter your results using these side filter options. But we're going to look at the gene page. So it's this first result here. And this is probably the page that you'll interact with the most if you're working with any sort of gene evolution or gene loss or any sort of the coding sequence. So this is sort of a summary of the gene. It's going to tell you if it's not named. Sometimes Ensembl will be missing names for genes. It will say protein coding or something generic like that. But some of them will have a big HGNC nomenclature here. But sometimes the description will actually have the correct gene name. So keep that in mind when you're searching. You might actually have to use the full length, the full name of the gene. And it tells you the location, so where it is on a scaffold in the genome. And it will summarize the transcripts for the gene. So there's 3 transcripts, 204 orthologs, or 204 genes in other species that Ensembl thinks is the same gene. And then 7 paralogs, so those are related genes within the species, right? There's some information about protein that I'm sort of like today. But so here's our transcript table. You can see how long they are, etc. Down here, I'm not going to show you because we're going to look at it in more detail. But just something to keep in mind, we're not Ensembl. If you're looking at these IDs, which always start with ENS, and then something specific to your species, and then either T or G. The T tells you that you're looking at the transcript model, which there may be multiple for any given gene, right? Because you have splice variants. And then the genes, this is going to be indicated by the G, and that tells you that that's the gene model. So that's sort of the home base for those transcripts. Okay, so we're gonna take a look at the Genome Browser. This is the way to visualize the genome, to visualize a gene in the context of this neighborhood, essentially. And here's what that landing page looks like. And it will automatically take you to the location of the gene that you started from on the gene page. So I know it's really small, but our estrogen receptor is right here. These red lines indicate where the gene model, the span of the gene model that Ensembl has predicted. And up here is the sort of like, this is the entire scaffold. So each of these red windows is zoomed in the next level. So this red window is what you're looking at down here. And there are all these tracks. And essentially these are all the models in this part of the genome. We're interested in these 3, which are all the estrogen receptor. And it is an important distinction between these top and bottom sections. The overview image is different from the region image. It's not just the same information, as you will see when I show you how to add things to what you can visualize up here. So you'll mostly probably be interacting with this region image. And then the navigation tools, which I'm sure you can all figure out, they're pretty straightforward. Just allow you to pan back and forth and to zoom in and zoom out on different regions, and they're basically the same between these 2. Okay, so I'm going to walk you through how to change what you can see, change how it's displayed, because you might have some data that's not in Ensembl, but you would like to visualize it in the context of this genome. So you might want to add your own track. So this is what it looks like when you open this configuration panel. You can see here it tells us that we're looking at the configuration for this overview image. There's also a region image that's separate. Again, just keep in mind they're 2 different panes. And I'm going to show you how to add a gene track that's found under genes and transcripts. This is already hosted on Ensembl, so this is not something that I had to upload. And you can see here there are 2 tracks that fall under this category of genes and transcripts. One of them is Ensembl. So Ensembl's already turned on its own track, right? Those red lines that we saw are Ensembl's gene models. But Ensembl is not the only maker of gene models. We've already talked about some other sources. NCBI is one of those, and they produce gene models that are called ref sets. Have you guys heard of a ref set or a reference sequence? Okay, so that's from another source, but we can actually add that NCBI-derived gene model set to what we're looking at. And so you do that by just clicking that little box and then choosing what kind of display you want. You can mess around with this on your own, decide what kinds of displays you like, with or without exons, things like that. I'm going to show you this sort of basic one. And then, well, like I said earlier, you can add your own custom tracks. You can talk to me about that later if you're curious. And now we have this extra track up here in this overview image that I've highlighted. And you can see they're blue bars, they're a little green with the yellow overlay. That's our RefSec track. So now we have 2 gene models mapped to the same genome, and we can compare between the 2. And then the same thing applies to this bottom region image. However, we have a lot more options to choose from under the region image track. So we have things like RNA-seq models, and we have repetitive elements, and we have conservation regions. There's a lot richer information on this lower, more zoomed in regional track. And I'm going to show you how to add the exact same one just for simplicity here. It's under genes. Again, we're going to go and click on this little box that's empty. We're going to turn on this RefSec track. And now we see that has been added here. And it's always going to add 2 tracks in Ensembl because Ensembl separates forward and reverse strands into 2 different lines, essentially. But this is now, you've represented the same thing that you got up here, but just in more detail. You can see the exon and intron structure. You can see if you have UTR. Thank you. Dang, that was fast. Okay, here we go. Strap in. We're going to just blow through this next part. Okay, so this is how you view the genome browser. We're going to skip over this. As I said before, there are both gene models and transcript models. They each have slightly different information attached to them. The gene model is going to have a lot more of these features. But you can look at things for the transcripts like protein domains. And I'm going to highlight some of these really quick. You can look at splice variants. You can download sequence, and that includes protein sequence, it includes mRNA, it includes UTRs. You can select all of these things for a given gene. And then there's comparative genomics information. You can get gene trees that Ensembl's already generated for you. You can look at orthologs. You can look at these genes in other species if they are in Ensemble's database, disclaimer. So Ensembl obviously has more than just Manakin in their database. And whatever genes they have that match the estrogen receptor in other species will show up here. Let's skip over this and talk about this later. This is important. But time. Okay, so paralogs, so related genes like estrogen receptor 2 will be in this list. And really important, you can see where the information came from to build these gene models by going to this supporting evidence for a gene. And this lets you see exactly where the evidence came from. So here we can see there's this ID here, same as the ID here, and then intron supporting evidence, which if you click on tells you it came from RNA-seq reads. So that's great, we have some coding sequence to support this. And then we can see what the aligned model was that helped build this gene model. We see that it's the zebrafinch ESR1. So, and just to contrast it with a genome that has a lot more information, this is what the evidence looks like for a zebrafinch gene. Okay, you can download the entire genome if you want by going to their FTP download site here. I'm pretty sure you can figure this out on your own. There's lots of links that take you to an FTP style site that lets you download them. And finally, Biomart. So Biomart is a way that you can download the whole genome. Maybe you have like a couple hundred genes that you want to look at. Biomart is how you get that data. So you can filter the Ensembl database to get exactly what you want. So there are different databases. Mostly we work with genes. Maybe eventually we'll have variation regulation sets for these genomes. So you choose genes, you choose the species you want data from. So in this case, I'm using the Manakin here. And then you're going to choose filters and attributes. And filters are your selection criteria. So that could be a chromosome that you're interested in. That could be only genes that have orthologs in the other Manakin. And the possibilities are endless in this part of Biomart. And then the attributes are the data that you want. So that might be coding sequence. That might be a flanking sequence. That might be more metadata about, like, maybe you want NCBI session values from the Ensembl model. So again, just check this out for yourself. There's so much data in this database that you have access to already. So to walk through an example, if I'm interested in getting flanking sequence, because I'm interested in sex-steroid receptor genes and how they're regulated, I can use Biomart to basically feed it that gene list and ask it to give me the flanking sequence that's upstream of the gene. So here's how you would do that. Under filters, you would choose gene as your filter, and you give it a list of genes. So I just typed in my 3 genes here. You might have a list of hundreds, you can upload a file. And Ensembl is going to, I can check to see that it found all 3 genes using this count function. Okay, it found all 3 genes, that's great. It's going to pull information for 3 genes. And this is the information that I want it to pull. So I'm selecting here that I want the flanking sequence. We're under the sequences attribute here. I want flanking sequence, and I want 2,000 base pairs upstream of all 3 of those genes. And then I hit results. We're going to skip through the, oh, sorry, one more thing. These are just more examples of data you can pull down. I added gene name here because I'd also like that to be in my header for the sequence pull. Hit results, and you get output like this. So This is just a preview. There's obviously more down below, but this is 2,000 base pairs for the 3 genes I asked for with the header information that I asked for as attributes. And then you can download that as a FASTA file. Moni already covered most of this, but just to sort of reiterate what she said, gene models are not always reliable, and that's due to a number of things: limited supporting evidence, so no transcriptome, or no sort of express sequence tags or things like that to support a model. There's gaps in the genome. There are fluctuations in genome quality and the sequence and the coverage, and they're often fragmented or incomplete models. This is a great example here that's actually right next to the estrogen receptor. In the Manakin, you can see that there are a bunch of models here where there should just be one. And I know that because Brevstat got it right. You can see here that there's a complete gene prediction. So neither of these predictors are perfect. So it's good to cross-reference using these tracks. And then finally, you might have genes that are not annotated or misannotated. So maybe Ensembl thought it was ESR2, but it's actually ESR1. So if you're requesting a list of genes based on gene names, Ensembl might give you the wrong gene, or it might say, I didn't find the gene because it's not annotated. And this is why curation is so crucial, because just being able to access the data is limited by whether or not there's a gene name sometimes. So you don't want to say the gene's missing just because it didn't come up in the search function. Just saying. The presentation will be posted, and these are some great resources you can dive into. Their documentation on Ensembl is wonderful. You can literally find these tutorials in video form and PDF form. It's great. Highly recommend it. You can also email me. You have my email information if you have questions. If you're like, how the heck do I do this? Like, I'm happy to help. I'll be around, obviously. And okay, that was it. Sorry if I went over.